This evening, we're very pleased to have Dr. Robin Polt. He's a professor in the Department of Chemistry and Biochemistry, and he also works with the Bio 5 Institute. So can we have a big round of applause for Dr. Polt?
Well, thank you very much for the introduction, and let's get started. We've we're at 3 talks now. This is the 3rd talk into this series, and so I'm assuming that everyone has already absorbed everything from the first 2 talks. So I'm going to not spend too much time recapitulating, but what I'm here to tell you about is taking neuropeptides or peptide neurotransmitters from the brain, and you can think of these as endogenous drugs. These are drugs that the brain produces to influence its own self, and the idea is from this we should be able to produce new drugs which could ultimately interact with the brain. The problem with neuropeptides as drugs in their native environment, they tend to just stick to membranes and they don't diffuse throughout the body, and so they're not very good drugs. And I'll speak a little bit about that, but mainly just give you the overall the picture from 30,000 feet. So the human brain's a fairly complex thing. There are 86 billion neurons or more, and in an equal number of non-neuronal cells that are all part of this thing. And of course, there are really big questions that people want to ask or answer. How are the neurons connected? How do they work? And of course, this is not something that's going to be figured out by one person or by a group of scientists in any short period of time. And various theories have been suggested. And I just want to preface everything that I'm going to say is that this is a chemist's point of view. And so our notion of how these things work is pretty straightforward here. There's just different parts to the brain that do different things. Now, there are, of course, a lot of issues with working with human brains, and people aren't interested in having things injected into their brains and so forth. So we tend to work with rodents, and we've heard from the speakers before the importance of the VT area, or ventral tegmental area, as well as the nucleus accumbens, which is the pleasure center. So we've talked a lot about dopaminergic and even glutaminergic transmission. Did we talk about GABA? I think we mentioned GABA as well. But today we're going to talk about opioids, which tend to be sort of a regulatory process for this. And the experiments that we do and our collaborators do are almost always done in rodents. And the fortunate thing is that our brains are not all that different from rats. And so you can just insert a rodent brain here for what we're doing. I want to introduce the topic by mentioning opiates. I think everybody is familiar with the notion of endorphins and runner's high and this sort of thing, and that's what we're talking about. But as a drug, opiates started with the poppy, the opium poppy. There is evidence that these things were cultivated, that is, opium poppies were cultivated well before the birth of Christ. The Sumerians referred to this as the joy plant, and it was believed that there were spirits in the plant that would inhabit your body if you consumed this plant. The Assyrians grew these things and taught the Egyptians how to do this. And of course, if you're not familiar, these drugs are derived from the latex that comes from the seed pod from the opiate plant, from the poppies. And the idea here is that if you scratch this pod, then this latex will will emerge, and there's opiates in there, essentially morphine. The Egyptians cultivated this thing, these plants, 1300 BC in Thebes, and these from there they basically spread all around the Mediterranean. The Phoenicians and Minoans traded in these things. We know that the opiates were there because we can read about this in the Odyssey. If you look at Homer's seminal work here, they talk about the lotus eaters that would consume some flowers that would make them forget about all their cares. And these were so delicious that those who ate of it left off caring about home. They didn't want to leave, right? Odysseus had to go and basically drag them back to the ships to bring them back to Greece. And so this was written about in the time of the Iliad and the Odyssey. Hippocrates, of course, dismissed the notion that there was anything magical about these things. He just said, no, this stuff's just a drug, and you just need to be careful about how you use it. And in fact, Galen described the first opiate overdose. I'm sure this wasn't the first opiate overdose, but it was the first description of it in the medical field. And eventually, the opium seeds were taken. The opium thebaceum is what they called them then. Was taken to China, 400 A.D. In Europe, Paracelsus was probably one of the first people to actually discuss this material. He called it laudanum, which is from to praise, and I guess it was something that was something worth praising for. And again, he said it was a very potent painkiller, but recommended that it be used quite sparingly. And finally, Friedrich Sertiner actually isolated morphine itself, which was actually the first alkaloid that was ever isolated in crystalline form. If we look at opiate poppies or opium poppies, typically called Papaver somniferum these days, there are actually several different varieties of this, some which produce morphine and some which produce thebaine. Thebaine, of course, coming from Thebes, from the Egyptian. And so the Egyptians actually knew something about these white opiates or white poppies, and people typically talk about good poppies and bad poppies because bad poppies produce morphine, good poppies produce mostly thebaine, which has no activity in it. If we look at what's in this sap, this latex, there are a number of transformations that occurs here. I'm not going to take you through sophomore organic chemistry, but these pieces come from amino acids, from phenylalanine, and eventually get coupled together and produce thebaine, which is sort of the end of the story if you're talking about the white or the pink poppies. But then these get converted on to morphine if you consider the red poppies. Now today we have ways of knowing what these molecules look like. Of course, you have to use your imagination to see these things because they're very tiny things, and you have to. There's literally no way to see them, so you have to imagine that this is the structure that is appropriate, and you can see it's sort of T-shaped, and we'll say a little bit about that later on. So thebaine and morphine, well-known alkaloids. Codeine was also isolated, which is a little less potent than its morphine cousin. Has this methyl group here. Interestingly enough, only about 85% of the people actually get any pain relief from codeine. Some people take codeine, they don't have the enzyme to remove this methyl group, and therefore they don't have any pain. There's no analgesia present because the only reason that codeine is an analgesic is that it gets converted morphine in the liver, and that's where your pain relief comes from. Thebaine, on the other hand, can be oxidized to form oxycodone, and apparently the Egyptians knew how to do this some 3,000 years ago, and we're just learning how. All of these plants produce apomorphine, which is an emetic. It causes you to vomit, which is apparently a useful thing to have. And if you were here last time, you'll notice dopamine is sort of embedded in this, and this is there's a rich source of alkaloids there. A number of companies that you're familiar with, drug companies, got their start making morphine. So Merck, Darmstadt, Germany, 1827, actually made morphine. Bayer aspirin, they not only produced morphine, they produced heroin. And in fact, if you go to England, heroin is a drug that you can actually get today if you need it. A number of alkaloids sort of peeled off after this. Quinine, if you like a gin and tonic, the tonic has quinine in there, and that's the stuff that glows under black light. If you've ever seen it that way. Nicotine, cocaine, caffeine, all alkaloids that can be subject to abuse. The American Civil War brought morphine to the fore. It's probably the first conflict where morphine was actually used in a large amount. And of course, the medical world was pretty primitive in those days. They still used leeches. Ether, chloroform were the typical sorts of things. In fact, ether is still used when I was a kid when they took my tonsils out. So there's actually quite a bit of opiates that were dispersed in these days. And the doctors, before they joined the army, whichever army they wanted to join, they were prescribing opiates already. They were giving either opium or morphine or some preparation. And I thought it was interesting. Bill Taylor here was a Confederate surgeon, and he noticed that he's just simplified the sick call to one basic question: are your bowels open, and do you have diarrhea, or do you have are you all stove up? And if you were stove up, he gave you a blue mass, which was a mercury compound that caused your guts to start working. And if they were running, then he gave you a plug of opium. On the Union side, there was a doctor that he actually performed a diagnosis from horseback, and he would just actually dispense morphine by sprinkling some on his hand and then letting the troops lick it off his hand. He didn't even have to get off his horse. And so things were not in great shape in those days. Of course, injection, the hypodermic syringe was invented about the same time, and there were huge amounts of morphine that were dispersed. On the southern side, there's actually, because of the blockade, there weren't too many opiates around, and so there was a great deal of suffering simply because of lack of pain medication. But on the other hand, there were a lot of people that abused this, and it was actually called soldier's disease or the army's disease. And I think that's a basic, if there's one point I want to drive home here, it's that opiate addiction is a physiological response to the drug. It is not, it's not necessarily because you have a poor well, it's a real thing. It's not just because you're a bad person. Even in the 1970s, there were claims that as many as 10 to 25% of the enlisted men in the U.S. Army were addicted to narcotics. Whether that's the correct number or not, I don't know, but it certainly was a serious problem. Some of the people that you may know, here's 3 generations of Lillies. Colonel Lilly, who came back from the war and saw this suffering and sought to alleviate it by starting a company. And the big innovation that Lilly brought to the field was sugar-coated pills. Before Eli Lilly and company, there was not sugar coating. And so this obviously has some positive things, but the companies that we see today that were around in these days selling opium, essentially, are the ones that invested in science and understanding what was going on. And you'll see that there were a lot of companies that didn't take this attitude. So we have Bayer producing heroin, Lilly producing heroin, and that was were the drugs that were used. You could even get morphine through the mail. If you thought you have a problem here, you can use morphine to cure morphine addiction. But at any rate, this was true. If your husband's a drunk, you can give him morphine. This one I really like. Tasteless discovery, which can be given in tea, coffee, or food, does its work so silently and surely while the devoted wife, sister, or daughter looks on. The drunkard is reclaimed, and even against his will and without his knowledge. So you just slip some morphine in there, and I could, perhaps, I don't know. But in any case, you were assured that it does no harm and causes no suffering whatsoever. Of course, as long as you take the drug. My favorite one of these was one-night cough syrup. You remember that one night? You remember that one night? That one night I took the cough syrup? No, I don't remember that either. And the reason I don't is because this concoction consisted of alcohol, cannabis, morphine, and chloroform, just to make doubly sure you got a good night's sleep. And these were skillfully combined with a number of other ingredients. So things were pretty bad before the Food and Drug Act. I have to tell you that. They really were. Steedman's powders was especially for children and cutting their teeth. And of course, there are actually a number of things that this is actually good for. And diarrhea, of course, will kill you. And opiates will stop diarrhea. And so I don't want to say that these drugs were not useful for some things, but they were certainly overused. Here, they want to make sure that you observe that this is Steedman's, because there's also a Stedman who sold opiates as well, but you want to buy the Steedman stuff. So there were lots of concoctions for children. Dr. Bull's cough syrup, which was just basically morphine in a syrup. One of my favorites was Winslow's soothing syrup. It will relieve all pain, relieves wind, which I think is flatulence, regulates the bowels. Of course, it shuts your bowels down, and 25 cents a bottle, not too bad. And Winslow's soothing syrup went on to be pretty good. And so companies that didn't invest in science, they invested in advertising. And of course, here's Mrs. Winslow's with her first kid, and she's got the little vial of opiates here. It's morphine, is what it is. And life got better for her. She's 2 kids, and she's still using the soothing syrup for teething. And eventually things got so well for Mrs. Winslow that she got a nanny to take care of the kids, and she didn't really have to do anything. Now, of course, there are side effects associated with drugs, right? So of course, these shut your bowels down, which is a good thing if you have diarrhea, but not such a good thing if you need to use, if you don't want to be incontinent, I guess. But at any rate, so Mrs. Winslow also sold a laxative to combat the side effects, and so this was great. You could sell the opiate to bind everything up, and then you sell the laxative to get things going. And so at some point you want to start using science to look at these things and try and understand what you're doing. And of course, if we take something like this, there's a plant, I guess, with some needles, and here's the same needles, And so you have to be careful. I mean, this is good. I mean, you can count how many sticks there are here and how many pine needles there are here, but what's order and what's chaos? Is this the orderly form, or is this the orderly form? We really don't understand enough about how life works to be able to answer all the questions. And so it's real easy to count pine needles, but what we really want to be able to do is to understand how they work together. So chemical structures are very important. We can actually, today's world, actually know what we're dealing with. This is morphine in its protonated form. And in the 70s, in the late 70s, people started discovering receptors. And so it took from 1977 till, about 2003 or so, where people actually started cloning the opiate receptors. They could actually look at these receptors, and this, we'll look at this more carefully. X-ray crystallography was important because we could actually get precise information about where these atoms were in the receptor and how the molecule fit in there. And a topic called molecular mechanics. What you're seeing here is a product of molecular mechanics, and in this particular case, it's a molecular operating environment. It's a commercial software package. So you can look at your opiate receptor. You can look at the ligand or pharmacophore. You can see here this green thing. This is bound in the receptor, and this is the part of this is the drug fitting into the receptor to cause something to happen. The receptor itself sits in a membrane, so this part here, you can imagine a bilayer going along, and so the outside of the cell and the inside of the cell, typically a nerve cell, the opiate binds in here, something happens, and you're allowed to feel something on the inside of the cell. We tend to look at these things in terms of their secondary structure, and peptides and proteins can form these helices, and so the opiate receptor, in some sense, we can just think of it as a bunch of helices, 7 of them as a matter of fact, that span the membrane, and then if the drug binds here, you get a little bit of movement among these helices, and then the G protein coupled receptor, the G protein is released, and the inside of the cell knows that something has happened. Here we can characterize this binding site. So it's there. This is just a molecular surface, and so you can see the green drug inserted in there, and then this purple and green surface here is the binding site, and we can look at that really closely, and we can look at it closer still. And so this little cave, if you will, is where the drug has to fit in order to get something to happen. So from a drug design point of view, you can just worry about this surface and forget about everything else. And so that little hole is where we want to put our drug or our binding. Morphine, again, we've seen this before. It looks like this. We can overlay that with oxycodone or OxyContin, if you would like the commercial name. And you see it overlays the hydroxyl here is about the same place the methoxy is here. You can see it overlays pretty closely. If we look at hydromorphone is Vicodin, I guess. And you can see they really all pretty much look the same with some difference. And for many years, people just started to tweak the system, and I would argue that it's like this sort of approach. There's a statement that people use in pharmacology. They often say methyl ethyl butyl futyl, and so you can go on changing little bits of the molecule, but it's still the same molecule more or less. But we can generalize this into the notion of a pharmacophore, which is just sort of a combination of several different moieties. The one that I showed you earlier uses this as a pharmacophore. This is not a real compound, right? This is just an overlay of several different opiate agonists. And if we look, we ask the machine, the Mo, say, just give us, don't show us all that stuff we don't understand. Just show us which residues, which amino acids on that receptor are interacting with the drug. And so here you can see aspartate 147. It's got a negative charge there, likes to coordinate with this positive charge. We've got some hydrogen bonding going on down here. And so this is how the thing works. But in the body, the endogenous opioids, the ones that your brain produces for itself, they look very different. They look very different. In fact, they're pieces of peptide. So if we look at dynorphin, for example, it's just a series of amino acids, but yet it binds to the same receptor. It doesn't look anything at all like morphine until you start looking very closely. I would argue that this is more like this. If you look at the drug, there's an N-terminus here. I'm calling this a drug. It's actually a neurotransmitter. But right there next to it is the aspartic acid, and you can get a hydrogen bond or a salt bridge going there. And then you have this aromatic group with a hydroxyl there. And then there's a place for another aromatic group over here that can engage in pi stacking. And so if we arrange these three moieties, these three pieces in the right geometrical shape, we'll get a pharmacophore. And that pharmacophore looks like this. The pharmacophore for morphine probably only fills up these two spots here. So this is a more comprehensive way of looking at this. And you can see here's our hydroxyl group, here's our hydroxyl group, here's the nitrogen with a positive charge, nitrogen with a positive charge. So morphine fits in here. Dynorphin takes up much more space. So here I'll show you some work from one of my students. Bobby Anglin actually was successful, got her PhD. There's a mu opiate receptor, and you can see this is actually an endorphin analog, and you can see this is stuck, the end of the peptide chain is stuck down in here into the receptor, and then there's this helical business up here that doesn't really seem to interact with the receptor in a normal sort of way. If we look at that maybe a little closer, you can see that. So this strand here, this white strand, is the drug, and then this blue mesh is the pocket or the binding site. And so we can get rid of all that and just think about how do we make compounds like this that we can bind into this moiety. Now, one of the big deals here is getting the drug from your syringe or your pill or whatever form you decide to administer it in to the site of action, which is in the brain. So this is where my colleagues, like Michael Hine, that you saw 2 months ago, talk about, do this sort of thing. And we can actually do microdialysis. We can actually insert insert little tubes into the mouse's brain. This is a rat, by the way, into the rodent's brain, and actually measure the drug in there by using his mass spec techniques. And I don't want to go through all that. But here's an example of a drug we made, a drug we call lactomorphin, because there's lactose. There's lactose over here, the disaccharide, our peptide, and that's an exogenous drug. And you can see here, this is evidence that the drug is in the brain. And here's an endogenous enkephalin, leucine enkephalin. And so this was already in there. So you can actually measure the drug that you've added as well as what you're doing, or as what's being expressed by the in this case a rat. Now we've got to cross the blood-brain barrier, and if we imagine that this layer of cells here, imagine this membrane, and we've got to get from the blood side to the brain side. What we think is going on here is that these drugs adhere to the membrane. They cause destabilization and invagination of the membrane, and then formation of these micelles, which eventually allow them to go to the other side and cross over. Now, what we do in our lab is we take these peptides and we put carbohydrates on them. And we can put a big carbohydrate, a medium carbohydrate, or a small carbohydrate. And what that really does is we're just affecting how much of the time does the drug spend on the membrane versus how much does it spend in solution. So it's going off and on, off and on. If I have a big water-soluble sugar on here, it tends to pull it into solution. If there's no sugar here, then it tends to stick to the membrane. And of course, that's great if you're a neurotransmitter. If you just have to go from one cell to another cell and they're only 100 angstroms apart, they can just sort of splat onto the surface, and that's good. They don't need to go anywhere else. But if you want a drug, it's got to go all the way through the bloodstream, through the liver, by the kidneys, and actually get to its site. So if it sticks to the first membrane it sees, that's the end of the story. And so by putting the carbohydrate on, we can get this thing to hop from membrane to membrane to membrane. If we put too much carbohydrate on, then it spends all its time in solution, and the receptors, of course, are in the membranes. We can look at these things using molecular mechanics. Here's what I've drawn here is the surface of the molecule in this blue and red mesh. And this is a compound called DAMGO. DAMGO is like the world's most fantastic mu opioid ligand. It sees a mu opiate receptor, it binds to it, and you get really great action out of that receptor. On the other hand, though, it's a terrible drug because if you inject it into your bloodstream, it just sticks to the first membrane it sees in the blood's in your circulatory system and never gets to the brain. So this business of having a lipid-soluble part or oil-soluble part and a water-soluble part, we call this amphipathicity. We give it an A value. And so a lot of the stuff we do, frankly, is pretty boring. And so I'm not going to bore you to death. But we can take very long peptides, and if they are long enough, they'll start to form a helix, and we can actually put water-soluble residues on one face, lipid-soluble residues on another face, something like this. So we actually get an amphipathic helix. Now this sticks to the membrane. What's nice about this is that this thing can unwind. And if this thing is not coiled up in a helix, it's water-soluble. So it can hop from a membrane-bound condition to a aqueous-bound condition. That's what I'm trying to say here, is that if this is very helical, it'll stick to the membrane, and then by putting sugars on it, we can pull it out. So if we have two sugars here, we tend to shift it this way. If there's only one sugar, it's shifted just a little bit. If there's no sugar, then we're stuck down here in the membrane. And it turns out Mother Nature's pretty good about this because it's already pre-designed all these things to have a helix with a fatty face on the bottom and a hydrophilic face on the top. It's almost like they've been reading my papers. And so, we're not, so we hop around. Eventually, we're going to hop onto a membrane that has the receptor of interest, and now we just search on that membrane in a two-dimensional context, and we can bind to the receptor, and we can get some action. And so we worry about how do we, how good is this binding, and how do we make it better, and this sort of thing. But one of the things we can do is we can simply measure analgesia after administering the drug in a normal fashion. So if we administer the drug in a normal fashion, intravenously, for example, then if we have a really good drug, we don't have to administer very much. We're way down here. If it's a very bad drug, we have to administer a whole lot to get an effect. So this is what we call the A50 value. So this is the amount of drug that it causes to get 50% analgesia. And the way this is typically done is by taking a mouse's tail and dipping it into hot water and seeing how long does it take him to flick his tail out of the water. So if that's very quick, you say, okay, that's a lousy analgesic. If the mouse doesn't care, that's a pretty good analgesic. And so that's the normal sort of effect. But we can also inject the drug ICV directly into the ventricular space of the brain through the skull. So we bypass the blood-brain barrier. And so this, if we measure this way, that's sort of a measure of the intrinsic analgesic ability of one of these drugs. If it were, if there were no blood-brain barrier, if we didn't have to worry about stability, then this would be the intrinsic value. Now, if we had the perfect drug, we'd be right here at the origin. We'd only need one molecule of drug to produce analgesia. We haven't gotten there yet, but we would regard drugs down here, which are intrinsically very analgesic and get through the blood-brain barrier and have lots of stability. These are drug candidates. And just to show you where we are, here's morphine over here. So you can do much better than morphine, fans. You really can, with better side effects as well. What kind of side effects are we talking about? Well, one is locomotor activity. And I'm sure you've all seen these old movies where the gangsters get a nag racehorse and they give it a shot of heroin and it runs the race of its lifetime and the 20-to-1 odd horse comes in. And that's not only because analgesia is caused by these drugs, but it's also caused increased locomotion. It causes them to move faster. So here's a 90 dose, which is well above the analgesic dose. And you can see this mouse is running in circles. He's got what's called a Straub tail. He's got the tail arched over his back, and he avoids this central space here. This is, in fact, this arched tail, this so-called Straub tail, is actually caused by dopamine effects. And so we've already heard about dopamine. On the other hand, we could administer lactomorphin here, same analgesic dose, and this mouse is engaged in normal sort of grooming behavior. And so the side effect profiles of these things are quite a bit different. Okay, so I could tell you more and more and more about opiates, but in fact there are many, many other neurotransmitters. Here's a few. And these are all G protein coupled receptor targets. There are about 700 or so of these in the genome, 250 that we know that are in the brain. And so here's the opioids over here. So you have three kinds of opiate receptor, mu, kappa, and delta that are listed there. But in principle, we could do lots of things here. Here's glucagon, controls sugar metabolism. Angiotensin controls blood pressure and other things. Vasopressin, oxytocin, the love drug. Okay, we'll hear more about that next time. But I want to tell you about a few of these. The ones we've looked at are angiotensins, opioids, and the so-called secretins down here, the PACAP, VIP1, and VIP2. If we start with angiotensin, so it's a little peptide. It's just 7 amino acids, but we can fold it in half, and the charged groups go up at the top, and you can see how the top part of this is very water soluble, the bottom part is very lipid soluble, so this wants to stick in the membrane. And so by putting sugars out here, we can pull it off the membrane, we can get membrane hopping, and very good activity. I can't tell you a whole lot about this project. This is actually Professor Meredith Hayes' project. And let me just say that the results are extremely good in the application that we're choosing on. Evan Jones is the guy who makes these glycosylated versions of this. But what happens when you inject these into the bloodstream? If we look at what happens in the bloodstream, here, this is minutes along the bottom here. You do the injection. Of course, you see a large bolus of compound going in. And then it very rapidly drops off. And by 20 minutes, you're down here to this, 40 minutes to here. And by an hour or so, the compound's virtually all gone. Why is that? Well, these compounds are rapidly being cleared by the kidneys. So the blood, of course, is circulating through the kidneys. Glycopeptides are removed. But that doesn't mean that the drug isn't going other places during that period of time. If we look at the stability of these things, if we look at angiotensin itself, and we just put it in rat serum, you can see it starts to degrade pretty rapidly. And the half-life, after 14 minutes, half of the compound is gone. After another 14 minutes, another half is gone, and so on. If we just convert the end of the peptide from an acid to an amide, I know this is organic chemistry, then you see it gets a little more stable. But when we put the carbohydrate, it's hours. It's really quite stable. And so this is good if you want to make a drug out of this thing. And we can actually measure the concentration of the drug at a small dose here in red, both in the serum, so in the bloodstream, as well as in the brain. These are these are after 20 minutes, I guess. And then if we use a larger dose, of course, there's more drug present and even more present in the CSF. That doesn't look too impressive till you realize this is, of course, a log scale. And if you go back to normal scale, you can see, of course, that you're getting quite a bit of your drug into the brain. And even though after 20 minutes, the drug has basically disappeared from the bloodstream. We know this drug is quite active because of NO production measurements. NO is a neurotransmitter that's, well, it's very important. It's the stuff Viagra interferes with, but also is useful in heart disease as well. But here's our drug here, and you can see it's very effective. Here's the native peptide, much less active. So we've looked at angiotensins, we looked at enkephalins, which is nature's pain reliever. Endorphins and dynorphins, of course, do other things besides relieve pain. They are, of course, analgesic. But what you see here is you have this same message that you find here is here. And now we've got this amphipathic helix attached to it. And then we've got this little linker. And that's all you have to do to remember about that. The helix, we're not the ones to invent this. E.T. Kaiser back in the 80s showed that if you look down beta endorphin, you'll see this helix. So where we're going with this is with a collaboration to cure Parkinson's. And the point here is we're not just treating the symptoms of Parkinson's. We hope to be able to treat the actual progression of the disease. That is, we want to rescue cells. So I can't do this by myself. All I can do is mix chemicals together and isolate them. But we've got Hein to study stability and blood-brain barrier transport. John Stryker looks at these drugs in cells and see this all takes just a little bit of drug. And then when we find what we think is a great combination here, we can turn these over to Torsten Falk, who actually looks at these in what are called Parkinsonized rats. So if we look at these receptors in a somewhat different way, so here's a map of all the G protein coupled receptors, and you can think of one receptor being created however many million years ago, and now it's evolved over time. And so we've been bouncing around over here with the opiates. Let's go somewhere else. Let's go up here to look at the secretins. And these are much older receptors. Pharmacologically, or I guess I should say evolutionarily, these are much, much older. And whereas the opiates control things like pain, attention, memory, the secretins control things like sugar metabolism, fat metabolism, oxygen levels, things that are really life or death, and they're actually much older than the opiates. They're much longer. Here's the single code letters for the peptides, and these are 27 or 28 amino acids. And the question is, does it work on peptides of that size? So PACAP is one of the drugs that we're interested in, or one of the neurotransmitters that we're interested in. And again, you have a message, you have a little bit of flexible linker here, and you have a helix. Nature's already done all this design work for us. And what we do is we simply attach a couple sugars at the end here in order to pull this thing out of the membrane so it can hop from membrane to membrane to membrane. PACAP interacts with what's called a class B GPCR, in which there's this big extracellular portion, and you can see how the helix fits into a groove on this extracellular portion. If we look at the opiate receptors, they don't have any of that. They just have this part. And so what happens here is that you bind to this groove, and then when that's bound properly, it closes down like a clamshell and shoves the message into the GPCR. A cartoon version of that is here. So you can see just binding to the transmembrane part of the receptor, or just binding to this part won't activate it. You've got to bind to both parts at the same time, and then you get activation of the receptors. One of the things that's pretty neat is we can just cut the head group off of these things, and then we can bind to the extracellular portion, and we can block the endogenous drugs that way. And so we can make either an agonist that turns the receptors on, or we make an antagonist that turns them off. And if we're smart about this, we'll be able to turn some on and another class off at the same time. So really the issues we've got are penetration of the blood-brain barrier, stability, both in the flask and in vivo, and then activity in vitro. So we've shown that we can get stability. My kind has shown that we can actually measure this drug in the brain after it's been administered. And here we just have a simple glucose here, and that's a mass spectrogram. And we see activities in cells that causes differentiation of these cells. So I think this is a fairly fertile field. If you look at the pharmacopoeia in the United States, fully more than a quarter of our drugs are directed towards GPCRs. And well, you can see that's a pretty good chunk of the action. And if you look at things over time, the number of peptides that are going into the clinic are getting greater and greater. And so I think in the future, we'll look back at the use of morphine and its metabolites as drugs the same way we look at sort of primitive plants being administered. You know, just here, take this plant preparation. It's got mystical properties, and I think we're gonna have much better drugs. And of course, I don't do any of this work. These guys do. Here's some people that have done this. This guy here just most recently got his PhD in November. He was going to stick around until August and helped train the next student. In fact, he went out and got a job. Can you believe that? He was tired of being a graduate student and making almost as much money as I do. I also have to thank National Institutes of Neuroscience and National Institute of Neurological Disease and Stroke, National Science Foundation, NIDA, Office of Naval Research, and thank you for your attention.